My brief today is very simple. I'm just going to give you a short introduction to how Ambroxol got to where it is now. And it began 150 years ago. So 150 years ago, a French physician called Philippe Gaucher described a disorder which presented most often in children, but also in adults, with a disorder that included enlargement of the liver and spleen. About 50 years after that, it was identified as a genetic disorder, and in the 1960s, it was identified as a disease due to the presence of two abnormal GBA1 genes, which produces the enzyme glucocerebrosidase. And these mutations in these Gaucher patients causes the accumulation of protein and fats within cells and therefore causes them to not work properly. It was a disease without any treatment to begin with. And then another 100 years from the original description of Philippe Gaucher, neurologists and people who began to work with Gaucher disease noticed it was rather unusual that people with this rare disorder seemed to develop Parkinson's disease more often than you might expect to by chance. And if we then move forward to 2009, there was a definitive paper which showed that mutations or changes in the GBA1 gene that cause Gaucher disease can also, when present only in a single gene, increase the risk of developing Parkinson's disease. At the same time, a laboratory in Toronto was looking at compounds that might be used for patients with Gaucher disease to increase the activity of this defective protein, glucocerebrosidase. So in 2009, there were two events which started this whole story. In 2012, we described that in patients who had died with Parkinson's disease, the glucocerebrosidase was deficient, both in those who carried a GBA1 gene and those who didn't. So we had the idea that maybe, given the results from Toronto, we could use Ambroxol to increase the activity of this defective enzyme in Parkinson's disease, and looked at this compound Ambroxol. Ambroxol, interestingly, had been around for many decades. It's been around since the 1960s as a cough linctus. So a very simple, very well tolerated, relatively cheap cough linctus that can be bought over the counter, and yet it has this fascinating ability to increase the activity of this enzyme that is defective in Parkinson's disease. So we thought we need to lay out a logical path to try and find out whether this compound is really going to be capable of helping people with Parkinson's disease. So this slide is a summary of the last 10 years' work. And I know you think 10 years is a long time, but this has followed a very logical progression which has led you up to the first clinical trial of Ambroxol, which was ASPro-PD, described in 2020. So that was only 5 years after we started, and Marco Toffoli is going to talk about that subsequently. But let me just tell you how we started out with Ambroxol. We started taking cultures of skin cells in the laboratory from people with Parkinson's disease and GBA mutations. And we put Ambroxol in with the cells, and we found that it significantly increased the enzyme activity. So that was a big positive result. We then wanted to ask, is it going to work in nerve cells? So we then took some so-called stem cells from people with Parkinson's disease, turned them into neurons, gave them Ambroxol, and sure enough, the Ambroxol not only increased the glucocerebrosidase activity, it also decreased the alpha-synuclein level that was accumulating in these neuronal cells. So this was a double benefit and very exciting. So we then started to move to in vivo experiments. We made, together with some people in Sheffield, a model fly of GBA deficiency. And we gave these flies, they drank it quite happily, Ambroxol, and their lifespan was increased. They did not lose their dopamine neurons, and they remained mobile throughout their life. So this was a really big result from this study. We then moved to Sheffield and the development of some fish. And these fish had an abnormal GBA1 gene, and we gave them Ambroxol, and they too had benefit in terms of increased glucocerebrosidase. We then moved to the mouse model of Gaucher or GBA deficiency. And sure enough, when we gave these mice Ambroxol, and they too drank it quite happily, we found that the Ambroxol increased the glucocerebrosidase activity in the mouse brain, and also, like in the neurons, reduced alpha-synuclein. So this means that we've gone through this whole series of experimental models to confirm that Ambroxol worked. And at least in the mouse, got into the mouse brain, because that's always an important question to ask, because you need to get the drug into the brain. Well, it got into the mouse brain, but we needed to know, would it get into a higher order animal brain? And so we did some tests on a monkey, and sure enough, the monkey brain had high glucocerebrosidase activity following Ambroxol. So we were now ready to move from the laboratory into the clinic with this drug which had shown so much promise in the laboratory but needed to be tested in people with Parkinson's disease. So I'm going to stop at that point and hand over to Marco.
Thank you. Right, so I join Professor Schapira in thanking the Cure Parkinson for the support for this research. And I must warn you that Professor Schapira's story started 100 years ago. Mine starts 2,000 years ago. I'll give you some more details on the story of Ambroxol and how it came to be. But first, I need to give you some boring information that is important to understand everything that will come later. So first, apology for being very basic, but what is a gene? What is GBA? So DNA, which we all have inside our cells, is divided into genes, and each gene produces a protein, which is something that our cells need to do a function. And if there is a change in the DNA inside the gene, the protein that is produced might be malfunctioning, might be misfolded, or might not be able to exert its function. And we call these variants, these changes in the DNA, as variants or mutations. We know we get one copy of each gene from our mother and one copy from our father. Now, if we talk about GBA, as Professor Schapira was saying, if we inherit two mutated copies of GBA, one from our father and one from our mother, we will get Gaucher disease. If we inherit only one copy, we will not get Gaucher disease, and we will be referred to as healthy carriers or non-manifesting carriers. However, non-manifesting carriers of GBA variants will have a slightly higher risk of developing Parkinson's later in life. And to give you some numbers, in the general population, less than 1% of people will carry a mutated copy of GBA. But if we take the whole of people with Parkinson's, around 12% of them will carry a GBA variant. And this is even higher in people of Ashkenazi Jewish ancestry. Moreover, the risk of developing Parkinson's, which in the general population can be estimated at around 3%, in people that carry a GBA variant is higher, is between 3 and 15%. So it's still relatively small. The majority of people with GBA variants will not get Parkinson's disease, but it is significantly higher. This, however, is very important because it allows us, now that we know that GBA is important, we can do something, as Professor Schapira was saying, to target this pathway and potentially alter the degeneration in Parkinson's disease. And again, this might be a bit too specific, but I wanted to give you an idea of what happens inside the cells of people that carry a GBA variant. The normal GBA gene will produce this enzyme called glucocerebrosidase, which you can see, you can see as the pink C-shaped object on your left. This will travel through the cell and reach the lysosomes, which is the yellow sphere on the right, and it will exert its function. And cells really need the lysosomes to work to wash away all the dirty laundry of the cell, all the things that the cell doesn't want. If you have a mutated GBA gene, it might produce a misfolded protein, which is this purple S-shaped object you see on the left. This protein will struggle to reach the lysosomes and will be eliminated somewhere else in the cell by that orange bean-shaped object that is the proteasome. And so this leads to two problems. The first one is that the lysosomes have less GCase, and so it's not working properly. And the other one is that the proteasome is degrading the misfolded glucocerebrosidase and doesn't have time to degrade other proteins. And this in turn leads to the accumulation of other proteins, including alpha-synuclein, which might be familiar, is the protein that accumulates in the neurons of people with Parkinson's disease. Now, the main questions surrounding GBA and Parkinson's disease are: who among the carriers will get Parkinson's? How is Parkinson's disease associated with GBA different from the rest of Parkinson's disease? And possibly the most important, can we do something about it? And the first two questions, what we are doing to try and answer is the two studies called PD Frontline and Rhapsody, and you will hear about it later. Now I will focus on the third question: what can we do about it? And the first concept I wanted to the last concept I wanted to mention before talking about Ambroxol is, in a way, it is a timely matter, because by the time people develop the motor symptoms of Parkinson's disease, the process is already underway. Neurons are already damaged. And so it's important to start the treatment sooner rather than later. Enter Ambroxol. So now let's move back to 2,000 years ago in India. If you developed a cough and you went to your doctor or your healer or priest, chances are they would have collected a herb called Adatoda basica, prepared a herbal tea, and give it to you. And this tea actually worked. What it did was to make your mucus more fluid, so it was easier for you to eliminate it and heal from a chest infection faster. And it's been around for very long. Then in the 1970s, some German doctors that traveled to India became very interested in this herbal tea. They started to study it and isolated the molecule, which was called Ambroxol. And since then, people have been treated for chest infections with Ambroxol worldwide. At the moment, it's not commercially available in the UK for this particular indication, but it is in most European countries and in the US. And as Professor Schapira was saying, the link with Parkinson's disease came in 2009. So some very clever researchers that were looking for molecules to study in relation to Parkinson's disease said, okay, if we start from scratch with a new molecule, we have to do everything: safety assessment, design the molecule, and this is a long and expensive process. So let's do the opposite. Let's take all the molecules, all the medications we already have, or thousands of them, we give them to cells that are GBA deficient and see what happens. And they did that, and they found that the cells that they gave Ambroxol to were thriving, they were working better, as you heard previously. And since we know that GBA is important not only for Gaucher disease, but for Parkinson's disease because of everything we just said, then this was very promising. And what we believe Ambroxol does, and the studies of course support this, is help the glucocerebrosidase to fold correctly and reach the lysosomes. So in this cartoon, Ambroxol is the gray, I'm sorry, I don't have a pointer, is that gray sphere that is binding to the purple S-shaped glucocerebrosidase. It makes it become a C-shaped glucocerebrosidase and reach the lysosomes. This process is called chaperoning. By doing this, the lysosomes are working properly, and the proteasome has time to degrade other proteins, like alpha-synuclein, for example. We like to think of ourselves as very clever, but other people had similar ideas. And there have been several clinical trials on Ambroxol going on around the world. Cure Parkinson's with Professor Schapira carried out the first phase 2 clinical trial with Ambroxol on Parkinson's disease, and we'll see what the results were. But there are others. There is one in Italy which is targeting Parkinson's disease associated with cognitive impairment. There is a large one targeting people with Lewy body dementia, which is a condition that is strictly correlated with Parkinson's disease, and so forth. So a few words on the trial we did, the phase 2 trial, and then you'll hear more about ASPro, which is the phase 3 clinical trial. So the trial involved 23 participants that were treated with high doses of Ambroxol for a period of half a year. And for that time, we assessed different things, including signs of toxicity and signs of Parkinson's disease. But the most important outcomes were that we were able to show that Ambroxol reached the brain, was able to cross that barrier that is called the blood-brain barrier, and reach the brain and engage with its target. There was a change in the cerebrospinal levels of glucocerebrosidase and even alpha-synuclein. The blood level of the activity of the glucocerebrosidase increased as well. So all of this to say, from the data obtained from the phase 2 trial, it is apparent that the Ambroxol can reach the brain and can engage with the right proteins in the brain. And most importantly, it was safe to use. Of course, if millions of people take this medication worldwide for a chest infection, we're already fairly happy that this is a safe medication to use, but the dose we are studying to treat Parkinson's disease is significantly higher. And even at these higher doses, there were few adverse events reported in the phase 2 trial, and these were all considered mild adverse events. There were no severe adverse events reported. We were all fairly happy with this, and this is why we decided to move on with the phase 3 clinical trial, which is ASPro-PD, and you will hear about it from Mairead. Thank you.
Good afternoon, everybody. My name's Mairead. I am the trial manager for the ASPro-PD trial, and that basically just means I'm responsible for its day-to-day management. You can see here that there are quite a few other people who are also involved in the trial. So we have our project manager, Felicia, Julia and Kashia, who are responsible for collecting and analysing all of the data in the trial, Marco and Eliza, who are our clinical fellows, and they provide all of the clinical support for the trial, and of course Prof Schapira, who's our chief investigator. So I've put our website on the slide here, so if you want to keep up to date with where we're at with ASPro, you can do that using our website. And for this afternoon, I thought I would talk a little bit about ASPro and how we got here. I also want to touch on what taking part in the trial is like for a participant. And then I'm also going to touch on the setup process, because as I'm sure many of you know, it took quite a long time to get to where we are. And that is because there is quite a lot involved in setting up a clinical trial. So ASPro-PD is a phase 3 UK multi-centred randomised placebo-controlled trial. Our plan is to open 15 NHS sites in the UK, and I'm pleased to say as of today we have 4 sites open. So the first site that we opened was UCLH in London. We then opened the Salford Royal, Fairfield General, which is also in the Manchester area, and then the Derriford Hospital, which is in Plymouth. So our participant target, so how many patients we need and want to recruit into the trial, is 330. And we have 24 months to recruit that amount of people. And that clock actually started last month when we recruited our first participant, and that was at UCLH. Because we started last month, that means that the recruitment for ASPro will end in February 2027, or sooner if we are lucky enough to meet our recruitment target before that time point. So we can see here the follow-up periods. That just means how long a participant is actually on the trial for. So it's a total of 34 months. The first 2 years are blinded. So that just means that neither the participant or their doctor will know whether they are receiving the Ambroxol or the placebo, so the dummy drug. Following that 2 years, all of the participants in ASPro will receive Ambroxol for 6 months, and we just refer to that as the open label phase of the trial. As Marco has explained in a lot of detail, the drug that we're investigating is Ambroxol, and the matching placebo is identical to the Ambroxol in its appearance and its texture. It just doesn't contain any of the active properties. We are also conducting a sub-study, so that's just like a smaller trial within a trial, and this will involve giving participants the option to have a lumbar puncture on 2 occasions, and that is for research purposes only. So what taking part actually looks like for a participant on the ASPro-PD trial is outlined here. So if somebody has thought that they may be potentially eligible for ASPro based on things like their age, time since diagnosis, and whether they've had their GBA sequencing done, they may be approached and invited to attend a hospital clinic where they will have a discussion, an in-depth discussion with a doctor and the research team about what the trial involves, any potential risks or any potential benefits. After this, if somebody would like to take part, they will complete what's called informed consent. So they will just sign a document that says that they are fully willing to take part in the trial and that they have a good understanding of the trial itself. This isn't the end. They will still need to go through the eligibility review. So the doctor and the research team will conduct a lot of assessments to make sure that this patient is eligible to take part. And the reason that we do this is to make sure that it's safe for that individual to be on the trial. If they are found to be eligible, they will be invited back to attend a baseline visit within 28 days. And it's at this visit they will be what we call randomized. So basically a computer will randomly assign them, so like flipping a coin, to either the Ambroxol arm or the placebo arm. The computer generates a number, so it's still not clear what arm they are to the patient and to their doctor, and that number is just matched up with the bottle in the site's pharmacy, and that's as much as they know at that point. So they will also have some symptoms assessed and complete a lot of questionnaires and be asked a lot of questions about their quality of life at that baseline visit, and then they can go home. And they will continue to take the Ambroxol or placebo at home for 5 months, and they will also continue with their usual Parkinson's medication as they normally would. Following that 5-month period, and on the next 4 occasions that they're invited to the hospital, we refer to these as visits. They will have those same assessments that they had at the baseline visit repeated again. And the timeline between these visits is generally about 5 months. When that participant then returns for their visit 6, they will enter the open label extension of the study, so they will be given Ambroxol at that point. And again, they will go home and continue to take that for 6 months until they return for their end of treatment visit. So at that end of treatment visit, they will stop taking the Ambroxol, and their time on the trial is essentially over, although if there is any issues, they can still contact the research team, and they will just continue to take their Parkinson's medication as they were already. Once all of the participants who have been recruited complete all of these visits and the data has been thoroughly reviewed, all of the participants who have taken part in the trial will be informed of its results. So that's the participant journey. Now I'm working backwards a little bit, but I'd like to just talk about the setup and how we actually got to the point of being open to recruitment, because it was quite a long process. The first step is to develop all of the study documents, and we always start this with the protocol. So the protocol basically is a very large document that comprises all aspects of the trial and governs its day-to-day management. There are a lot of people who are involved in writing this protocol. It goes through several review committees, and it takes a long time to get it right and get it to that finalized state. Once we have that finalized state, we use that protocol to inform all of the other documents that we require for the trial. So we have an IMP management document. So this is all of the documents which detail any information that site pharmacies may require about the Ambroxol or the placebo, such as how they order it, how do they store it, how do they dispense it, and any kind of safety information that the doctors at the site may need. We also have to write several safety management documents, and these outline how we will monitor safety throughout the entirety of the trial and the process for reporting any urgent safety measures to the regulatory authorities. Sample management. So as part of the trial, we do collect a lot of different samples from the participants, which can include things like blood and CSF, So we have a lot of plans which outline how these are collected and how they are processed by the teams at sites, and then how they are analysed by our laboratory team afterwards. All trials have to undergo quality assurance, and this is done on an ongoing basis, and just ensures that all of the critical data points are being collected and that the participants are being monitored for any safety events. The electronic database is designed by the trial team, and then it is built by our in-house programmers, after which it undergoes several months of testing to make sure that it works correctly, and then we will give the teams at sites permission to use it and to enter real-time data. Finally, the participant-facing documents. So this includes things like the participant information sheet, an invitation letter, or a poster, all of the documents that potential participants will see. And we worked very closely with people with Parkinson's to develop these documents and make sure that they are clear for the people who will actually be receiving them. So in parallel with writing the study documents, we also need to think about selecting the sites. And there's quite a lot of thought that goes into the selection of these sites. So we wanted to make sure that we had a good spread of sites across the UK, so that as many people as possible would have access to the trial. We need to make sure that the site has the capacity to run the trial to an excellent standard, and that they have all of the equipment that they need to run the trial to the protocol. We also need to make sure that they have enough potential participants in that area around that hospital site so that they are able to actually recruit. Next, we need to identify vendors, and by vendors I just mean any third party who is supplying a service to the ASPro-PD trial. And the most important one, arguably, is the IMP vendor. So we need to make sure that the company that's manufacturing the Ambroxol and its placebo meets all of the regulatory requirements and has all of the appropriate licenses in place. We conduct an audit and review all of their standard operating procedures, and we need to make sure that we have all the systems in place to make sure that the drug can be shipped in a secure and timely manner directly to the sites. And then all of this is kind of comprised into a contract which needs to be reviewed by the legal bodies before finally being signed. And we also have this same process with all of our laboratories who are doing sample analysis, so it does take some time. So once we have all of the documents, we have all of the sites that we think we're going to have in the trial, and we have contracts with all of our vendors, we can then initiate the ethics review and trial approval process. So the MHRA review the pharmaceutical and scientific aspect of the trial. The ethics review involves a board of both experts and lay people, and they review the trial to make sure that it is ethically sound and that the benefits outweigh the risks for potential participants. And then finally, the Health Research Authority will review all of this information, and they can either refuse the trial, make recommendations, or approve the trial, which is thankfully what we had for ASPro-PD towards the end of last year. Once we have that approval, we are then able to approach sites' research and development departments, and they conduct their own internal reviews of all of the documents just to make sure that they have everything that they need at that site to conduct the trial. We then need to set up contracts and discuss finances with each of those sites. They will then need to be supplied with all of the appropriate equipment and documents that they need to actually run the trial. And we also provide training to every single member of site staff that will be working on the trial. Once we have all of that, then we are able to green light that site, which means that they will be able to open to recruitment. So in terms of where we are at the moment, as I mentioned, we've got 4 sites that are open to recruitment: UCLH, Salford, Fairfield and Derriford. As of today, we have screened 4 participants and 1 has been randomised. Hopefully tomorrow that will be 2. And the plan for the next few months is to continue to open those sites in a staggered manner throughout the rest of the year. And then the next steps will be to start with that interim data analysis. So at different time points throughout the trial, we want to look at the data and make sure that the safety data is sound and that it's safe for the participants to continue on the trial. So just some of the key points about who can take part. You will need to be between 35 and 75 years old, have a confirmed GBA status, have been diagnosed within the last 7 years, and not have had a deep brain stimulation. But as I mentioned, there are quite a few other eligibility criteria which are further assessed at hospital sites by the doctor and the research team. For anyone who is interested in taking part, I would advise you register with PD Frontline. The PD Frontline team are going to speak a little bit more this afternoon, and also discuss it with your doctor. And if you have any further questions, you can email the trial team on our trial inbox, which is listed here. I think that is it from me. Thank you very much for your time.